Hi students, today we're going to talk about DNA and the central dogma of genetics. DNA is made up of three components. You have your 5-carbon sugar or deoxyribose sugar, you have your phosphate group which make the rails of the double helix ladder, and then you have your nitrogenous bases which make up the steps of the double helix ladder. The nitrogenous bases are like bonded together by hydrogen bonds. The nitrogenous bases are adenine, thymine, cytosine, and guanine. Adenine pairs up with thymine, so A pairs up with T. Cytosine pairs up with guanine, or C pairs up with G. So these make up the sequence of the nitrogenous bases, determine what you're going to look like. So for example, I might have A in a certain region where you have T, C, G, A, C, okay? And this might translate to making proteins that have melanin. Okay, melanin is the protein that makes your hair color darker. So if you have more melanin like me, you have really dark hair. And if you have a lack of melanin in your hair pigment, then you have lighter hair than I do. So, okay, so the way it all links together is you start off with DNA. You get half of your genes from your mom and half of your genes from your dad. Okay, your DNA is unique to you, your sequences are unique to you because it's not entirely your mom's and it's not entirely your dad's. It's different. So what happens is it starts off with one strand of DNA and you have to make many copies just because you have many cells. It started off with one fertilized cell, but now we have a lot of cells that we need to have that same DNA in. And DNA is found in the nucleus, and it stays in the nucleus. So you need to make different cells and different DNA, so you have to start off with replication. So the way DNA replicates itself is it uses 2 original strands to make 2 new strands, and in turn 2 DNAs. So let's say this is DNA. Okay, you have your 5' strand to 3', and your 3' to 5'. Okay, so when replication starts to occur, it opens up into a replication fork, and you get an entire new strand, hook up with this strand of DNA. So you have an old and a new, okay, and now you have one separate DNA. And on this side, you still have that old strand, and then you get a new complementary strand hooking up with that DNA. Now you have an entire different 2 DNAs, although they have the same sequence. So that is replication. So replication, DNA makes more DNA using 2 old strands and making new strands. That is what we call semi-conservative. So you're always going to have one old strand and one new strand in any DNA molecule. After the DNA has replicated, it needs to transcribe into the RNA. Because the DNA cannot leave the nucleus, it needs RNA to go in there and transcribe the information to carry out to the ribosome and make proteins. So in transcription, you have your DNA and it opens up like a bubble so the RNA, which is single-stranded, can go in and get that information and take it to the ribosome. The one thing about RNA is it stands for ribonucleic acid. It's slightly different than DNA because, again, it's single-stranded. Instead of thymine, it has uracil. The way I remember it is if I could be DNA or RNA, I'd choose RNA because it has U in it. I know it's cheesy, but it reminds me of why RNA is different than DNA. So RNA transcribes, that's transcription. So DNA makes RNA in transcription, and then RNA goes into the ribosome of the cell and it creates proteins, and this process is called translation. So in order to create proteins, we need to look at the RNA and break it up into codons. So 3 letters makes up one codon. So A, and then you go to the different codon, C, G, A, and then different codon. Okay, so that is how, depending on what codon you have, that is the protein that is going to be there. So again, you have your codons, and then you're going to have your anticodons that pair up with your codons. So for example, if I have A, my anticodon would be U, and it carries a certain protein that goes with it. So just quick summary. You go from one strand of DNA to a lot of strands of DNA through replication, then DNA to RNA through transcription, then RNA to proteins through translation. I hope that helped you guys just refresh a little bit about DNA and central dogma. Thanks.